Hello. My name is Jake. I am a medical doctor taking care of critically ill patients with COVID-19, and I want to transmit to you the success we are having in the management of them in the intensive care unit. I want to teach you what I know, what you really need to know. There is a lot of information outside, not everything helps. I will teach you what it really helps in few minutes. I will guide you through 10 short videos about the basics. The medicine essentials. Let's start. First, we are going to talk about what is coronavirus. I will explain you about its origin, groups of coronaviruses, the structure of the virus, and the replication cycle. Let's begin with the origin of severe acute respiratory syndrome (CoV-2). Let's begin with the origin of severe acute respiratory syndrome (CoV-2). Everything started with the first patient with symptoms on December 8, 2019. In that day no one knew a new virus was the etiology of that. Atypical pneumonia. On December 31, 2019, several local health facilities reported clusters of pneumonia of unknown etiology that were epidemiologically linked to a large seafood and live animal market in Wuhan, Hubei Province, China. After this discovery, the market was shut down on January 1, 2020. On January 7, 2020, a coronavirus called severe acute respiratory syndrome (CoV-2), initially named as 2019 novel coronavirus, was officially identified as the cause of an outbreak of viral pneumonia. 4 days after that, on January 11, first death in China is recorded. The first case outside China was on January 13 in Philippines, and on January 23 Wuhan was placed under quarantine. On February 11, World Health Organization indicated that the name of the disease caused by novel coronavirus is COVID-19. Co means corona, VI virus, and D makes reference to disease. 19 is for the year of its discovering, 2019. The World Health Organization recognized the disease as a pandemia on March 11, 2020. More than 2 months after China alerted about the pneumonia cases. Before we continue describing the severe acute respiratory syndrome (CoV-2), we have to talk about the different groups of coronaviruses. Coronaviruses belongs to the subfamily Coronaviridae which are further classified into 4 groups by phylogenetic clustering. The groups are the alpha, beta, gamma, and delta coronaviruses, which alpha and beta are known to cause infection in humans. The first human coronavirus was identified in the 1960s, and it was classified in group. Until 2003, only 2 human coronavirus species, human coronavirus 229A and human coronavirus OC43, were recognized. Currently, 7 different coronaviruses strains are known to infect humans, including 229A, NL63, OC43, and HKU1, which generally cause self-resolving infection. There are also severe acute respiratory syndrome coronaviruses, known as severe acute respiratory syndrome coronavirus, SARS-CoV-1. Middle East Respiratory Syndrome Coronavirus, MERS-CoV, and newly identified severe acute respiratory syndrome (CoV-2), SARS-CoV-2, which can cause lethal respiratory infections in humans. This is a comparison of the 3 coronaviruses known to cause severe viral pneumonia. Severe acute respiratory syndrome coronavirus 2 has 89% nucleotide identity with bat severe acute respiratory syndrome, like coronaviruses, and 96% with that human severe acute respiratory syndrome coronavirus discovered on 2003. The severe acute respiratory syndrome coronavirus 2 is less genetically similar to MERS-CoV, around 50% nucleotide identity. Severe acute respiratory syndrome coronavirus and severe acute respiratory syndrome coronavirus 2 use angiotensin converting enzyme 2 as the receptor to enter the host cell. MERS-CoV uses a different receptor. The incubation period for 3 viruses are similar. The estimated R0 of severe acute respiratory syndrome coronavirus 2 is between 2 and 3, which means that each person with the infection is expected to infect 2 to 3 other people in a susceptible population. For comparison purposes, the average R0 for seasonal influenza is around 1.3. The severe acute respiratory syndrome coronavirus 2 infection primarily affects adults, with fewer cases reported in children of 15 years or younger. The overall fatality rate varies by location, intensity of transmission, and variations of care. The nationwide mortality rate in China has been around 4%, 5.8% in Wuhan, 1% other areas in China. In China, the overall fatality rate was higher in the early stages of the outbreak, and has decreased over time to 0.7% for patients with symptom onset after February 1, 2020, which could possibly be owing to evolution in patient standard of care. However, the mortality rate is higher in Italy, Spain, and United Kingdom, while it is much lower in South Korea, about 1%. It is not known whether these differences are due to higher percentage of older patients infected in those countries, widespread testing in South Korea that increases the denominator by including more asymptomatic or mildly symptomatic low-risk patients, or other undetermined factors. But let's emphasize about the severe acute respiratory syndrome coronavirus 2, known as SARS-CoV-2, causing COVID-19. It belongs to the beta-CoVs group. It causes a severe respiratory illness with many epidemiologic, clinical, radiologic, and laboratory findings similar to severe acute respiratory syndrome coronavirus infection in 2003. Transmission seems to be primarily from person to person via close contact, through respiratory droplets, with a mean incubation period of 5.2 days, ranges from 2 to 14 days. There is some concern about a possible fecal-oral route of transmission for severe acute respiratory syndrome coronavirus 2 because patients with severe acute respiratory syndrome and MERS frequently had diarrhea, and the virus RNA was detected in stools of patients with severe acute respiratory syndrome coronavirus. Gastrointestinal symptoms, such as diarrhea, abdominal pain, and vomiting, have been reported in 2% to 10% of patients with COVID-19, and a March 2020 report from a U.S. patient showed positive stool test for coronavirus, but there is no absolute evidence of that route of transmission. I will talk about the routes of transmission in another video. Structure. You have to pay close attention to the structure of the severe acute respiratory syndrome coronavirus 2. This is the base to understand about the replication cycle and the pathogenesis. Coronaviruses are named for crown-like spikes on their surface. Coronaviruses are single-stranded positive sense RNA viruses, with the capacity for rapid mutation and recombination. Coronaviruses are known to cause respiratory or intestinal infections in humans and animals. They are large pleomorphic spherical particles with bulbous surface projections. The average diameter of the virus particles is around 120 nanometers. Coronaviruses contain 4 major structural proteins: the S protein, the spike, which mediates attachment to the host receptor, and subsequent fusion of the virus and cell membrane, the N protein, the nucleocapsid, the membrane is the M protein, and the envelope, the E protein. The viral envelope, with a diameter of 80 nanometers, consists of a lipid bilayer where the structural proteins are anchored. A subset of coronaviruses also have a shorter spike, like surface protein called hemagglutinesterase, the HE protein. It is a protein with enzymatic activity that binds to the sialic acid of mucosal cells, cutting it off. It is a dimer that facilitates detachment of the virus from the sialic acid-rich epithelium. Inside the envelope, there is the nucleocapsid, which are bound to the positive sense single-stranded RNA genome. The lipid bilayer envelope, membrane proteins, and nucleocapsid protect the virus when it's outside the host cell. Now let's talk about the replication cycle. It is divided in 4 steps. First step, receptor binding. Coronaviruses have a large single positive RNA-stranded genome, making it the largest RNA genome of the RNA virus family, enclosed in a nucleocapsid of helical symmetry. These viruses infect human cells via S protein binding to receptors on host cells, followed by release of viral RNA into the cell cytoplasm. The S protein of SARS coronavirus 2 binds to ACE2, a transmembrane receptor which is widely expressed in lung, heart, kidney, and gastrointestinal tissue. Second step, entry. Following receptor binding, the virus must then access the cell cytoplasm. This is assisted by acid-dependent proteolytic cleavage of S protein, by a cathepsin, or another protease, followed by fusion of the viral and cellular membranes. S protein cleavage occurs at 2 sites, within the S2 portion of the protein, with the first cleavage important for separating the receptor binding domain and fusion domains of the S protein, and the second for exposing the fusion peptide. Fusion generally occurs within acidified endosomes, but some coronaviruses can fuse at the plasma membrane. Third step, replication. Following the entry of the virus, the viral replicase gene, which consists of 2 open reading frames coding for 2 polyproteins. These polyproteins contain all the non-structural proteins of the virus which are essential for intracellular replication. Then, the virion releases its RNA. Some RNA is translated into proteins by the cell's machinery. Some of these proteins form a replication complex to make more RNA. Many of these proteins then assemble into the replicase transcriptase complex, which replicates the viral RNA. Following replication and viral RNA synthesis, the viral structural proteins S, E, and M are translated and inserted into the endoplasmic reticulum. These proteins move into the endoplasmic reticulum called the intermediate compartment, where viral genomes encapsulated by N protein bud into membranes containing viral structural proteins, forming mature virions. Fourth step, release. The viral progeny are finally transported by Golgi vesicle to the cell membrane, and are exocytosed into the extracellular space, where the new virus will found another cell, to infect and repeat the cycle. This is enough for the first video. Thank you very much for being here with me. Go to the next video. I will be talking about epidemiology. I will teach you about the transmission of the COVID-19 and many other things.